Please go ahead.
Thank you. So good afternoon, colleagues, and it's great to be here, and thanks for the invitation. It's a real pleasure to be here and speaking with such a diverse audience. I was looking through the screen and seeing many persons from science and regulations and colleagues from other countries. So thank you. The good part for me is really that we're all working on different parts of the same puzzle, which is trying to improve the implementation of a Phage platform for Europe. So over the past years, I think Phages have shifted from something which was anecdotal, experimental, and to something that now sits in the European strategic conversations about biotechnology and health security. And that's really what my presentation is about, is how can we move from recognition, which is very recently, to regulation and really impact. From promising science to an actual framework that brings phages and phage therapies to patients safely, quickly, and in a sustainable way. So I would like to walk you through what has changed very recently in the past 2 years, and especially since we've been engaging also with PhageEU and the partners, and what we are helping to build in terms of the new architecture that we're proposing for the regulatory framework for phages in Europe. So but let me just walk you through a bit of myself and LxBio. So basically, before we explore the policy landscape, I would like to introduce you LxBio, which is the organization that I'm affiliated with. So we're a Lisbon-based biotechnology company, which is currently it's focused on next-generation biologics in 3 verticals. So one is obviously phage therapy. Second is antibody engineering and regenerative and precision medicine. So we're actually looking also into areas like nanoparticles and using nanoparticles also for enclosure of antibodies. And we also provide CRO services for small and medium companies and for biotech companies. So our current team, has combined, professionals combining about 20-25 years in industry, in biotech and clinical development, regulatory affairs, and industrial applications. I come from a space which is mostly on the market access part and the regulatory and HTA technology evaluation. So our mission is quite simple. It's really to develop biologics and that transform infectious, inflammatory, and degenerative disease. We're also looking into oncology applications mostly on the antibodies part. This is, because we're engaged in these platforms, in particular in the phage area, we do have a couple of technologies we are very, coming to fruition. One of them is we're concluding the phase 2 trial. Actually, the last patient, last visit for the phase 2 diabetic foot infections phage cocktail is expected this month, in the month of December. And we will likely have, announcements to make throughout the first quarter next year. And this gives us, a very peculiar place to be, where we are getting closer to a proper regulatory approved therapy. So we're getting very close to it. But looking now into the thing that brought us here is really when just recently in last year we heard Mario Draghi speaking about the future of European competitiveness. It's really one of the the message that he really delivered to us was really that innovation is not a subsidy, it's a capability, and it's really it's what we aim to discuss here is really that lens is fully applied to phages. Because especially in biotechnologies is really about capability and how would we enable Europe to turn scientific knowledge into this strategic autonomy. if we keep just only discovery, but not only, it's not just about discovery, it's about production, regulation, adaptation of biologics within a proper, adequate biologic framework in Europe. And so in the particular case of phages, this is a perfect illustration of that, because phages are really biological entities. They're very precise, they are very adaptable, and they evolve and they do evolve. So they're not just chemical compounds that you can simply, automatically reproduce in a factory. So they adapt. And this is as immediate regulatory implications. So the success will depend on well we match the bacteria to match each phage and phage cocktail to the bacteria that we want to control. And this creates a very different regulatory and manufacturing challenge comparing to traditional antibiotics. So adaptation is key here. Meanwhile, I think when we look at Europe, Europe is starting to redefine its frameworks, but others are already, testing theirs, namely US and Canada. They are already running adaptative trials and hospital phage programs that are running for years under very flexible investigational rules. They are moving very quickly, not recklessly, but quickly. They are experimentally, and they are learning by doing. And Europe risks falling behind, not in science, because we do produce excellent science in the field of phages, but in the speed of translation, meaning how we adapt the regulations and the frameworks to make it to the patients. And the longer we wait, the more likely is that clinical data, manufacturing know-how, investment will consolidate elsewhere. So that's a call for priority. So when we say phages without permission, is really, we're not talking about rebellion or something unregulated. We're describing a decade of responsible clinical work that happened, some years ago, that by necessity was moving faster than the law. So clinicians.
Excuse me, Luis. Have you been changing slides? Because I am still on the second.
No. I'm moving.
Okay, that's good. Just wanted to be sure. Okay, please continue. Sorry.
Yeah. So basically, this is a key point. Really, is patients and clinicians were treating already. Clinicians were already treating patients, and Europe was not adapting its own framework. So this is really what we need to address. So if we look in the last, decade, I think we can see that this curve tells the story better than any documents. For over a decade, from 2010 to 2023, phage therapy was lived in a gray zone. It was really a purely national reality. We had Belgium, Poland, France. Each one was building their own measures or protocols, starting with Belgium. But they were, pragmatic, they were functional, but they were isolated. They were mostly about individual magistral formulations on a named patient basis. But this phase did matter because clinicians proved that phages could be delivered safely, responsibly, even without an EU framework. And that experience gave us credibility to what is now following. Then we see in 2024, the curve begins to rise. It coincides with also the birth of the PhageEU. And by April, we do have the pharma package, the pharmaceutical legislation consultation introduces the concept of adaptive biologics into EU legislation. It opened up, this possibility to discuss the role and place of phages. By June 2024, phages reached the European Parliament as part of the antibiotic resistance framework and industrial strategy. It was the first time that we did see a mention of phages as a possibility, as an option to fight antibiotic resistance. And by December 2024, the Common Declaration of MEPs recognized the role and importance of phages. And this was done across very different political groups from different political families. By 2025, the slope is intensifying, it steepens. We had just very recently engaged as PhageEU in an AMR dialogue in June this year, where we did discuss a lot of topics related to phage and phage adoptions and the regulatory framework. I would say that the three key points were really one about the agreement or at least in principle to explore pilot modular authorizations. We will go through that in a minute. Meaning modular authorization for phage cocktails as a and as a approved medicines. By also in 2025, we started also to have discussions with on the Biotech Act. It's still ongoing process of consultation, and there's more to pending for next year. And by 2026, we do expect to have more news about the Commission proposals on the Biotech Act. But in this part, there was already discussions about biobank infrastructure support, more proportionality around the support given to small and medium companies and biotech companies, which are in essence the majority of the developers in the phage space. And recently, the Innovation Act also providing additional support in the infrastructure part and helping to build up the pilots in the adaptive pathways for the phage technology. So in essence, in 18 months, we did see a lot of change, and we can see that the discussions are happening right now. And they will be for the most part, coincidental in time in terms of what we should be expecting over the period from 2026 to 2028. Let me go to one more point, which is really the big policy documents that are already shaping this area. The first one is really between 2018 and 2020, Belgium created the first national framework for phages. They use existing pharmacy law, classified phages as APIs, as active pharmaceutical ingredients for magistral use, for individualized use. And they published a monograph defining purity, characterization, and manufacturing standards. So this is not GLP standards, but it still addresses the need for a common framework for quality. This was a quite revolution because this was the first time phages were recognized under a serious proper regulatory oversight, even though national. And between 21 and 22, France and Poland adopt similar approaches. And there was already, by 2023, there was already a network of hospital pharmacies in those three countries. And very recently in my country, Portugal also adapted a similar framework of personalized treatment through the magistral route. And that that evidence is now giving Europe, a lot of, cues about what Europe needs to advance. And very recently, we did have, following a regulation on the veterinary side in 2023. Just recently, EMA publishes a draft guideline, which is under consultation, and it's which already treats phages as a biological medicinal product approved under the Medicine Statute of Directive 201/83. So this is the first time that we see, an official paper by EMA formalizing phages as a biological medicine therapy. So basically, Belgium built a foundation EMA is now scaling this, even though for different routes, one personalized, and this one is more for the centralized approval of phage-based products, but EMA is now scaling this up to the wider EU. When EMA released its initial concept paper, which is now back in 2023, what was really the point is there was kind of a very specific phrase that what they were recognizing that there was 27 languages, meaning that there was 27 different ways to approve phage therapy under a personalized treatment. That was true that every country had its own recipe, its own paperwork, its own definitions, and locally it worked, but cross borders was not, it failed. Basically, there was no major collaboration between countries. By October 2025, the draft guideline, which is now the one that we are discussing, it makes a very different decisive move. It defines phage therapies as biological medicinal products and puts phages alongside with vaccines and antibodies like legitimate biologics, not exceptions. So this is no longer an exceptional use. It's being positioned as other biologics. This guideline is now currently focusing on manufacturing, characterization, stability, control, and it applies only to strictly liquid phages only, so no genetic modified, and references or the European Pharmacopoeia. So it's not the final system, but it gives Europe a shared grammar for centrally approved products, how to describe a phage, how to test it, how to control it. So, and without a shared grammar, I think you would never create cross-border access. I mean, that's still missing. So let's just look a bit deeper on the current EMA guideline. So we see EMA, the draft guideline gets a lot of things right, meaning definitions, GMP baselines, alignment with the pharmacopoeia, clarity on the lytic versus temperate phages and GMOs. These are really the solid foundations, real progress. And but as foundations, they are not the full building. So it's the foundations. We still missing the upper floors, which are more related to access, to adaptability, and to evidence. And basically, I will sum that what is still missing, and which will make a reality of phages in Europe, is really in the case of access, we still miss adaptive or modular authorization for phage cocktails. That means that every single strain change that's required due to to bacterial resistance or need for more effectiveness. Every time we change a phage within a cocktail, this is currently being treated as a full product change, and this is biologically is unrealistic. This is one of the first things that we need to address. Second one is that it doesn't connect in any way, short of form. There's no connection between the centralized approval process and Article 5.501, which is basically related to hospital and inpatient use, the measures of formula. And there's, one of the obvious parts is that we could learn through real-world evidence, data collection also, to get evidence which would support the application for the centralized approved products. And also on the infrastructure is really, there's still no reference to phage biobank infrastructure or network, and that's more what we address here is, the vision of having common standards and interchange, scientific collaboration between biobanks across Europe using similar standard definitions. So there's still that part. So I would call this the triad, which is, the process which are ongoing and not there. None of these are finished. They're under consultation and they're in very different steps, but this is the right time for us to discuss. In the pharmaceutical package, the delegated acts for strain variation, the one that I just mentioned, the incentives for, classifying phages to be eligible for AMR incentives, which are being done also for antibiotics. This is a missed opportunity, in my opinion. We could do much more in terms of incentivizing the development of phages for antibiotic-resistant infections, and also the data collection for real-world evidence for the data package to be used in the centralized approved products. On the Biotech Act is, the adaptive biologic dossier. It's more related to also the support of making sure that the requirements are proportional to the size and reality of companies and hospitals who are developing these technologies. So making sure that they are fit for purpose, but they are recognizing that the developers within this space are not huge, resourceful organizations. There needs to be proportional support for these companies, and support also for the phage biobank infrastructure. And the Innovation Act also is related to the funding elements and the support for the sandbox trials within when I mean sandbox trials is the adaptive part on the EMA side to support new regulatory pathways and experimental ways to start approving adaptive biologics. So funding must accompany flexibility, and that's our key message. Trying to tie together regulation, infrastructure, and investment in this triad. That's they have to be moving in the same direction together. So, and we're getting close to my, the finish of my presentation. But if I was to summarize our policy stance in these areas, is in one side, this would be one of this would be the one. These three, gears are that will lead regulation to move at the pace of modern biology. One is the pathway, that we should provide a proportionate, modular, adaptive biologic dossier, which is predictable and affordable for SMEs to develop these technologies. On the platform side is really ensure that there is a capacity and a possibility of doing modular authorization of phage variants. Again, the issue around strain change variation. This would require under and particularly under the pharma package regulation, that there is a pathway we propose a predefined proportionate pathway, which would be a type 1 variation equivalent, which would be based on comparability, meaning a framework whereby we predefine, similar to what's happening right now with vaccines. We do define the platform by which the phages are prepared, and by switching only the phage and keeping all the rest constant, we would be able to, to have a simplified variation approved. In practice, meaning more on the magistral side, that we come together sharing, common data standards. No, it's not centralized approval, but at least having harmonized magistral baselines by which, a phage which is developed in Antwerp or Lyon could be delivered in Lisbon and following the same technical standards. So it's more as a federation than rather a centralized approval. That's what we are proposing here. So, and this would be a very distinct European model, which still allows a lot of implementation and collaboration between states, if they so wish, but sharing a similar, scientific infrastructure. So my last slide is really about, looking at the outlook and giving you a couple of examples what could happen, from now until 2027 scene. But my key point is really in that triad of events, the pharmaceutical legislation, the Biotech Act, they're all pretty much happening right now, pretty much in the same space and at the same time. So it could be possible by 2026 that we will have already delegated acts or the design of delegated acts allowing for modular adaptation of Phage in the centralized approval, at least the proposals, and a pilot could happen pretty much throughout 2027. It's very possible that upon the Commission proposal of the Biotech Act, establishing already some kind of some level of support for the biobank infrastructure, that we could start in parallel already to set up some kind of infrastructure of collaboration network across Europe, you know, building on examples from other areas like in the vaccines area that we, and the biobank network already happening in Europe, that we share the common idea of having same data standards and same scientific requirements for a wider European phase network and collaboration. And my key message here is really, if Europe can regulate phages at the speed they are evolving, recognizing they do evolve, and we need to find flexible, adaptable ways to implement. We have learned to regulate the future in itself. And basically, I think phages have this capacity of being in the right time and space to provide examples for other areas, like for other areas where adaptive you know, management of technology, especially in biotechnology, will happen anyway. So it provides a safe ground with already enough you know, practice and support for us to pilot in this particular space. And this is my last slide. Thank you very much for listening.
Thank you, Luis. Indeed, a lot of information. Thank you for this complex summary of more or less what is happening currently at EU level. Indeed, as PhageEU, we see this increased interest into phages as AMR is rising, and we as PhageEU, we make sure that indeed those initiatives that you have mentioned somehow cover also phages. A lot of congratulations for your presentation. So indeed, it was relevant. So as I said, PhageEU is really active in terms of advocacy to make sure that indeed phages are covered by those initiatives. Maybe an interesting information for all of you is that PhageEU has been invited by the European Parliament to speak during the Public Health Committee on the 8th of December during a hearing about innovations in AMR. So it will be another occasion for us to indeed convey the messages that you have just presented, what is missing indeed in this future planned regulatory framework. I think another important message for everybody who is present here is to take part in the consultations of the EMA draft guidelines. Those consultations are running, I think, till April 2026. So still a lot of time, but we really encourage you to be active, because when I look also at the questions, most probably we don't know all the answers because this is now in the preparation. So if you wish to see something, if you wish to have something, it's really time now to take part in different ongoing public consultations. There is a number of questions, Luis, for you. If you could stop sharing your screen, and I will try maybe to share some of the questions that I have already noted. Otherwise, I don't know if I can. Okay, you stop sharing, and I will try to share the questions that we have received so far. Do you see them well? Are they readable, or I should make them bigger? It's okay.
Yeah.
So the first question is regarding the access point you mentioned of the European Medicines Agency guideline. What role will BRCs and lab phage collections play? Will just phages submitted to BRCs accepted, and what about the financial support? I don't know if you're able somehow to address this question.
Can you please clarify what was the acronym here? BRCs mean?
Okay, we can move to the next one. And if indeed in the chat box you could explain a bit the abbreviation, that would be great. The second question is about the sandboxes. Does sandbox mean sandbox for approvals of novel drugs or sandbox for regulators to create more regulation?
No, a sandbox is something which is already foreseen. It's really and it's under the you know the plan for the EMA plan for the next few years is given that new areas are by nature experimental. It's intended to give a more controlled, flexible way of regulating you know new drugs. It's applied mostly for new drugs, and because and in some of the cases we are approving new technologies and new concepts, and if we currently, in the case of Phages is one particular example, it's basically it's a supervised control environment where we do experiment in regulations. So meaning that we in this case here, and in our case, in our example, is if we explore the possibility of having a simplified variation approval of phage variations, that would be the place to implement. So it's a controlled regulation whereby we learn and then before expanding and make it part of the regulation. It also can apply, for instance, for using data package which are not fully as you know, for instance, incorporate real-world evidence in the evidence package, which is to be submitted for a centralized approval. You know, the so-called adaptive pathways, meaning that there is the possibility that under controlled environment we approved a medicine in a more a not so standard way. So it's more experimental.
The next question is about the ethical clearance. On the ethical clearance, do phage study needs any special approval apart from the apart from the normal clearance we usually obtain?
Yeah, this is very. It depends really on the type of study. But if it's the traditional clinical development for a medicinal product, is pretty much the same. It follows the same pathway as a normal drug or a normal biologics. There is one particular peculiarity of phages, which is by definition, it's not very feasible to do a typical phase 1 trial, because phages need bacteria to amplify. If there is no bacteria there, there's no way to do a phase 1, which basically we cannot experiment with or having a control arm with healthy subjects. But all in all, the process and the approval process is pretty much the same.
I think we have some clarification for the first question. BRC, it's biological resource centers, like ATC and CTC.
So basically my point, what I was trying to hear, there is no specific mention right now on the EMA guideline regarding phage collections. What they really, what's currently on the EMA draft is really technical specifications on the requirements for production and quality control. So this is extensively described there, and it connects directly with the European Pharmacopoeia. So the point here, what I was trying to mention is more about the biobanks and the capacity. If we do come up together, different biobanks across Europe that standardize the specific technicalities by which, you know, we map the genomes of the phages and the host bacteria. We do have common scientific specifications. We can have a place where it's still a national competence. It's still named patient-based use, but the interchange of scientific information in common standards could apply and could potentiate a more cross-border collaboration. That was my point, and that would be quite an, and this would in the future would also help generating additional information which could potentially be used for centralized approvals, meaning the utilization of evidence generated with hospital formulations later down the road for different approvals in centralized approved products.
Okay, the next question is, how does genetic modification of lytic phages fit into the plant framework?
It's quite simple, it doesn't right now. I think the option here was to, to start on a very clear, clean cut, so meaning genetic modified phages currently would be fall into a different regulation, and basically they will be treated as advanced therapy medicinal products, ATMPs. So it's they are managed in a separate. There's actually no case yet, but right now the draft guideline specifically excludes genetic modified phages from this designation of advanced therapeutic medicine phages.
Mm-hmm. The next question, number 5. It is important to have a risk-based approach to quality, especially GMP requirements. I will read it. Low bioburden products and a less complex production environment must be possible. Topical and intestinal applications do not require expensive sterile manufacturing.
Well, so let me clarify. I'm not a scientist, so here I'm more on the regulatory part and the access part. Anyway, my point on this one, I fully agree. And actually, right now, the requirements for a personalized treatment under magister formulation does not require GMP. So it does require some level of compliance with a quality standard, manufacturing standard, like in the case of Belgium. And actually, we did copy, by the way, in Portugal, we did copy and created a fairly similar approach to this, by which we do follow the manufacturing standards set by the European Pharmacopoeia and the Belgium Belgium got the name here, but the standard quality standard here. So there is this common platform which defines the minimum requirements for the production, and I mean, because it's less, it's usually developed for a specific patient, that it's no longer GMP is not required.
Okay. Number 6, I don't know if you are able to address it somehow. What is your view on the regulatory challenges of non-medical applications, ranging from biomanufacturing to environmental regulation?
Yeah, so here I prefer to defer a bit because I'm not a specialist and it's not my field of work. I do know that the key concern here is really containment. So all the issues around containment and exposure and ensuring that but I think it's probably there's probably someone in the audience that would better address this one.
Indeed, if there is anybody in the call who feels confident to respond to these questions, please indeed try to respond also in the chat and have some exchange. I think question number 7 indeed refers to question number 5 that we have addressed.
Mhm.
Question number 8 is again about the genetically modified phages. I don't know if it's something you are able to address or it's too specific. Not sure
It's too specific, but my inclination is yes, and there is a whole string of what you consider to the genetic modification. So, you know, probably someone on the scientific side could also help me on this point. But I think that the question is amplification and re-amplification of the same phage and trying to improve the genetic profile of a phage, which is called phage training, can be classified. It's actually it's not a genetic modification, but my inclination on this one is that it could be considered a genetic modified phage, yes, but this is now entering in a space which I prefer to defer to my science colleagues here.
Indeed. The next question is about any potential biobank. So indeed, if anybody has some answer, please respond. You may also try to get into direct contact if you see that you can somehow accommodate the needs. I think these were all the questions asked so far. I think there is still last one here. Okay, let me maybe stop sharing the screen. Let's have a final look at the chat. And then we will move to the next presentation. I see somebody has responded to this question about the genetical modification, so that's great. There is a question, maybe the last one, Luis, where does pre-adapting phages fall into these guidelines? Do I understand it correctly that environmentally isolated phages are what is registered? And even if they are just pre-exposed to pathogenic strains and mutated, they would need to be registered again?
Yes, they will currently, yes, and that's exactly my point initially on allowing us to basically what we want with this. Well, the what we learn actually from other disease areas like in vaccinology is that if you provide a proper framework for the changes to be done in a controlled way, in our case is the technology, the host bacteria, the process, the quality controls, everything stays the same. And the only thing that changes is there is one phage which is replaced for a phage within the same family and with a description that allows the regulator to understand this is a phage within the same technology, within the same family. It addresses the same type of pathogens. Then if all the rest stays the same, this is even though we do register as a modification, this is recorded as a modification. We follow this with pharmacovigilance on the safety side to make sure these variations are followed through. But essentially, it doesn't require for us to do the full development process. At this point in time, this is one area which is basically impeding or making it extremely hard for a company, for an entrepreneur to invest long-term, because once you reach the final stages of clinical development, if there is a need to change the phage, it has to do a lot of work again. And there is no sound scientific requirement to do such a thing. And that's our key point.
Last question. It's a short one. I'm not sure the response is short. Would you suggest the EMA guidelines only for the products for market authorization or for API as well?
The thing is, I think it's one of the areas we want to have both options open, meaning a centralized process by which you develop phage therapies standard, and which is it's a centralized EMA-approved product, is a phage therapy product. The other route is the exceptional route and in a magistral formulation. This is not currently within this regulation. It shouldn't be, although there should be somewhere a connection by which we learn from the local processes. And our recommendation is really that there is a scientific convergence in terms of what would be the requirements in each and every country. And actually, in practice, that is already what's happening, like in the case of France, of Belgium, in our case in Portugal. So we're trying to follow up similar manufacturing and quality standards when we choose the magistral route. And this is important for us to, actually to be able to, receive phages from different parts of Europe, because the key point here is really exceptionality, and it's a very specific case of a specific patient. So by which we need to be capable of sharing that information, making sure that and the biobanks also will help tremendously, because by then they're using, and there is probably my colleague from Phage, Precision Phage will shed some light in there, but by having common descriptors, we might, fasten the way by which we identify the proper Phage or Phage combo that's needed for a specific patient. And by sharing common scientific and production standards, the localization of this could be extremely helpful. So do different routes, EMA for centralized approvals, decentralized approval under Article 5.1, which is foreseen in the medicine statute, and some kind of connection, but still giving the freedom for the countries to collaborate by themselves, setting common standards. That's my takeaway.
Right. Thank you, Luis. I think you can rest now for a while. Please keep an eye on the chat.